Okay, everybody. Good morning. We're recording this on Friday the 13th. Dum dum dum. Don't worry. Last time we had Friday the 13th was back in March when we went all into quarantine. So what could happen? Today we're going to go over the stages of meiosis. And we're going to do meiosis versus mitosis and look at the differences between the two cell replication processes. Okay, so quick recap. Remember that meiosis is the division of one diploid parent cell into four haploid genetically unique, which means they are not identical, daughter cells. This is gametogenesis. So meiosis is the means in which gametogenesis occurs. It is the formation of sperm and it is the formation of egg. Now in meiosis, there are two rounds of cell division. In round one, you go from diploid to haploid. Oops. This is called meiosis 1. And it involves prophase 1, metaphase 1, anaphase 1, telophase 1, and you can call this next one interkinesis, or you may call it cytokinesis 1. Okay, now when you have those two daughter cells, they're going to go through a second round of division and result in four haploid daughter cells. This is known as meiosis 2. This includes prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis. And you can call it 2 if you want to. I'm not going to mark you off if you do. All right. Now, to understand the stages of meiosis, we're going to just go through the whole thing. And what we're going to do is we're going to do the meiosis of a animal cell. We're going to go through all the stages. I'm going to draw them out for you and explain what's happening in each one. So you have to go through all this. Okay, so let's begin. Well, you're going to start with interphase. And in interphase, you have G1, the organelle is double. You have S phase, the DNA doubles. And you have G2, which is when you make your final prep. And what we're going to do is we are going to do a fruit fly again, because fruit flies have only 4 chromosomes. Hold on. Yeah, we're only going to do a fruit fly because they only have 4 chromosomes, so it's going to make it actually really easy for us to draw this out. If we try to do a human, we would have to do 46, and then when it all doubles, 92 chromosomes, which is just madness. So we're not going to do that. So let's draw our cell. And we have our nucleus, everything's intact. We are going to have 2 centriole, or two pairs of centrioles now, which are called the centrosome because in G1, everything doubled. Okay, so let's indicate that. We have two centrosomes now because in G1, everything doubled. And also, let's say that this is a chromosome 1 from the parent, the male parent, chromosome 1 from the female parent, chromosome 2 from the male parent, chromosome 2 from the female parent. Now, I understand that in interphase, the DNA is not visible. I am doing this so that you can see what has actually happened. So this is before S phase, and then after S phase, you're going to have a copy of each chromosome. Okay? They're all going to be copied. So technically, you have 8 here, but you just have 2 copies of each of the 4 chromosomes. All right? Let's move on. Let's go into prophase 1. In prophase 1, the centrioles are starting to move to opposite ends of the cell, and they're starting to assemble the spindle. So we're going to write everything down. Centrioles move or migrate to opposite poles and begin to assemble the spindle fibers. Okay? The nucleus is beginning to erode. And the coating of the nucleus is called the nuclear envelope. So I'm going to say the nuclear envelope is breaking down. Now, here's probably the most important part, everyone. The homologous chromosomes. You should remember what those are. If you don't recall what homologous chromosomes are, refer to the previous video. Homologous chromosomes are going to go through crossing over. So chromosome 1 from the father is going to be mingling with chromosome 1 from the mother, and they're going to be experiencing crossing over, which means pieces of the paternal chromosome are going to be exchanged with pieces of the maternal chromosome. So this is going to be called crossing over. Crossing over occurs when homologous chromosomes pair during synapsis. So there's some vocab words here, guys, from the last video. You got crossing over, you got homologous chromosomes, you have synapsis. If you don't know what those are, be sure you go back to the previous video and learn that. Okay. And also the DNA is condensing. So obviously, the chromosomes are now chromatids. Okay. Let's move on to metaphase 2. Or excuse me, metaphase 1. I don't know why I said that. All right. Here's our cell. Here's one pair of centrioles. Here's another pair of centrioles. And I want you to really pay attention to this, okay? Watch this. Okay, here's what I want you to zoom in on. Look at this. You see this chromosome right here? Don't highlight that. That is a paternal chromosome. Let's just say blue represents male and red represents female. That's a paternal chromosome, which is now a sister chromatid, but it has a piece of the maternal chromosome on it, which means that it is an exchange of alleles for the same traits. So let's say that the father has long wings and the mother has short wings. Those are both the same characteristic. Those are wings. And you just have different forms of that characteristic, just like with eye color with humans. Eye color is the trait, but then you can have blue eyes, green eyes, brown eyes. And so this is what happens in crossing over. Pieces of homologous chromosomes exchange information with each other. And what this does is that it increases variation in the offspring. It increases variation in the offspring. Okay? Homologous chromosomes, or excuse me, homologous pairs align independently at the metaphase plates. Now, it is very important that you realize how this is different than mitosis. And when we get to comparing meiosis to mitosis, you'll see how in mitosis, it's all just one straight line along the metaphase plate. But here in metaphase 1 of meiosis 1, they're side by side. This is something new. Okay, let's continue to anaphase 1. All right, here the cell is starting to spread apart. So we have this one chromosome here. A little chunk there. I'm using all my blue when I need it. Okay. So in this stage, homologous pairs separate and move towards opposite poles. So very similar to what's happening in just anaphase of mitosis. They're being pulled to opposite poles. But notice in mitosis, you are pulling chromosomes apart. Here, you have an entire sister chromatid. So that's a little bit of a change up. Okay, next we have telophase 1. Starting to form that cleavage furrow. The spindle has essentially dissipated. You have a nuclear envelope starting to reform. And inside, you're going to have non-homologous pairs. Okay, so here we go. The spindle fibers have dissipated. Nuclear envelopes reform around the chromosomes. The cleavage furrow is evident. You're about ready to divide into two daughter cells. And then lastly, we have interkinesis. And we now have two daughter cells. Okay. Now, it's important to remember that this daughter cell is haploid. So its haploid number is 2, and this one is haploid. When we started, over here, it was diploid, and it was at 4 chromosomes. But now we're already at haploid. If you guys just compare, you had 1, 2, 3, 4 chromosomes. This is after I drew. If you rewind the video, you'll see that we had 1, 2, 3, 4. I'll highlight them and then unhighlight them. You had 1, 2, 3, 4 chromosomes. But over here, now you just have 1, 2, 1, 2. So you are already haploid. You just have two copies of the same chromosome. Okay. So next, you go back into interphase. Let's do meiosis 2 now. In interphase, you go through everything again. I'm not going to draw it this time, but you have the G1, you have S, you have G2. All that happens again. So keep this in mind. You double the centrioles again. You double the organelles, and I just want to be sure you all are aware that includes the centrioles. Okay? So let's get into prophase 2 now. So we have to double everything because we have 2 daughter cells that we're going to be dealing with. Prophase 2. All right. It's really the same old, just so you now have 2 cells. You see the nucleus is beginning to erode, starting to go away. And the spindles are starting to assemble again. All right, so let's draw the DNA inside. All right, so cells have 1 chromosome from each homologous pair node. Spindle begins to assemble. The nuclear envelope begins to break down. DNA is visible. Okay, let's continue. Metaphase 2. All right, this one should be pretty standard. The chromosomes align along the metaphase plate. All right, anaphase 2. You should know what's happening here. It's starting to be pulled apart. All right, here we go. Oh, hold on, I messed up on a little chromosome up here. Didn't I? Didn't have the little red. There we go. All right, there's one chromosome. There's the other being pulled apart. And then down here, get that one. And mostly maternal with a little bit of paternal on there. That's being pulled apart. We go down to the other ones. That's being pulled apart. There you go. Sister chromatids separate and become daughter chromosomes. Okay, let's get over to telophase 2. You have a nuclear envelope starting to reform. Spindle fiber is basically gone. And within each chromosome, you're going to have, or excuse me, within each nucleus, you're going to have chromosomes. All right. In telophase 2, the spindle disappears. Nuclei form. Cleavage furrow is evident or visible. Then finally, in cytokinesis, you have 4 haploid daughter cells. They're all haploid. Meiosis results in 4 haploid daughter cells. Okay, all right, well, that is all of, look at that. This is actually not that bad of a drawing there, guys, when you look at it from up high. We went over everything that happens in meiosis 1 and in meiosis 2. So I asked you this question on the last video. I want to ask you again. Which of the 2 stages of meiosis looks more like mitosis? Does meiosis 1 look more like mitosis? Or does meiosis 2 look more like mitosis? Think back to what mitosis looks like. To answer that, let's actually compare them. Okay? So this is going to be the second part of your video. This is going to be meiosis compared to mitosis. All right, so let's have a little comparative T-chart. Okay, let's begin. Meiosis requires 2 cell divisions. Mitosis requires 1 cell division. Just one difference. Next, meiosis results in 4 daughter cells. In mitosis, mitosis results in 2 daughter cells. Okay, in meiosis, the daughter cells are not genetically identical to the, genetically identical to the parent cell or each other. And in mitosis, the daughter cells are identical to the parent cell and to each other. All right, another one. Whoops. Meiosis produces haploid cells. Mitosis produces diploid cells. Okay, we got one more. Meiosis only occurs in sexually reproducing organisms to produce gametes, and that is called gametogenesis. Mitosis is more common because it occurs in all tissues during growth, repair, and replacement. Okay, so actually, let's draw it out, shall we? All right. So here's what all right, well, I'll just do it with you guys. But this is going to be abbreviated, okay? I'm not going to be going, what the heck, why not? All right, so I'll draw it with you. We'll do meiosis 1, meiosis 2, and then mitosis. Here we go. Prophase 1. You have your parent cell. Nucleus is eroding. Spindle is starting to assemble. And the DNA is going through crossing over. They're exchanging all their information. So in this one, synapsis and crossing over occurs. Okay. Metaphase 1. All right. So you got a maternal and a paternal chromosome that weren't really touched. And you have some that were. So you can see the crossing over that happened right here in the bottom too. That's crossing over. Homologous chromosomes separate, align independently at the metaphase plate. The reason I say independently is if you recall from last lesson that all of your traits are independent of each other. Not one trait is dependent on another trait. Just because you have brown hair doesn't mean you have to have brown eyes. So every trait is independent. It does not depend on the other. Okay, next we have anaphase 1. Chromosomes are getting pulled apart, or the chromatids being pulled apart. Okay, it says anaphase 1. Let's move on to telophase 1. You got a nuclear envelope starting to reform. Okay. So telophase 1, daughter cells are forming. And we'll go on to divide again. Okay. And then you have interkinesis. You have two new nuclei. All right. Okay, so that is all meiosis 1. Once again, there's all the stages. Now let's do meiosis 2. All right, we have prophase 2. The nucleus is starting to break down. You still have those sister chromatids. Okay, let's go to metaphase 2. Whoops. Okay, that's metaphase 2. They line up along the metaphase plate. Anaphase 2, I know this is tedious, guys. Just, we all have to go through it once. Okay, there's anaphase 2. We have telophase 2. Nucleus is starting to reform. And then we have cytokinesis. All right. So there's meiosis 1, meiosis 2. Now let's do mitosis. Stuff we did last unit. So this is just prophase. Remember, there's no prophase 1 or prophase 2. It's just prophase. Nucleus is eroding. You're starting to see the chromosomes. 1, 2, there they are. 3, 4, okay? Metaphase. Oop, I should probably draw the spindle starting to assemble, shouldn't I? There you go. Easy enough, right? Form along the metaphase plate. Anaphase. Can pulled apart. Telophase. New nucleus is starting to form. And then you have cytokinesis. All right, so you tell me. Look at the chromosomes. Just look at the chromosomes. Which two are most similar? Is meiosis 1 like mitosis, or is meiosis 2 look more like mitosis? The correct answer is mitosis has more in common with meiosis 2. Because as you can see, look at metaphase of mitosis. It's a single file of sister chromatids along the metaphase plate. Look at metaphase 2 of mitosis 2, a single file of chromosomes along the metaphase plates. All right. So let's compare meiosis 1 to mitosis, and then meiosis 2 to mitosis, and that'll be it for today. Here we go. Meiosis 1 and mitosis. In prophase 1, you have the pairing of homologous chromosomes. This is called synapsis. And you have crossing over occurs. In just regular old prophase, there's no pairing of chromosomes. They are not in pairs. Okay. Metaphase 2. Bivalence at the metaphase plates. Remember, they're side by side. And in just plain old metaphase of mitosis, duplicated chromosomes at the metaphase plates. We call duplicated chromosomes sister chromatids. All right, anaphase 2, or excuse me, anaphase 1. I don't know why I keep doing that. And I did it above, didn't I? I don't know what's going on. Did I do it on? Okay. In anaphase 2, homologs I spelled that a little wrong. Of each bivalent separate and duplicated chromosomes move to poles. In anaphase, sister chromatids separate becoming daughter chromosomes that move to the poles. And then lastly, we have telophase 1. Two haploid daughter cells not identical to the parent cell are formed. And then in telophase, you have two diploid daughter cells, and they are identical to the parent cell. Okay? Now, why don't we do this? How about this, guys? Let's make our lives just a little bit easier. Let's go ahead and throw in meiosis 2 in there. So let's say meiosis 1 and 2. And we'll put meiosis 2 over here so we don't have to rewrite everything with mitosis again, because that's what I was originally going to do. So let's compare, let's do prophase 2. In prophase 2, there's no pairing of chromosomes. In metaphase 2, you have a haploid number of duplicated chromosomes at the metaphase plate. Anaphase 2, sister chromatids separate. Becoming daughter chromosomes. Remember, the daughter just means the result of a parent. That move to the poles. And then lastly, almost done, we have telophase 2, You have 4 haploid daughter cells, and they are not genetically identical. Okay, so remember that meiosis 2 is more like mitosis than meiosis 1 is. That's something that you're often asked. All right, well, that is it. After this video, we are 1/3 through this chapter. In the next ones, we'll get through sections 5 and 6. In section 5, we'll talk about the human life cycle, which we actually did before the hurricane came. And then we will go over issues where there are where the chromosomes are not divided evenly. And so you end up having an individual with 1 extra chromosome, like Down syndrome, or one too few chromosomes, like Turner syndrome. So you'll learn all these various syndromes where an individual, a human, has an extra chromosome, or they have a missing chromosome. Okay? But that will be next time. I'll talk to you guys then. See ya.